What's the gap called? What are these circles called? Vesicles. What's in them? Neurotransmitters. If this were a muscle and this were a nerve, what's the neurotransmitter? Acetylcholine always. It's the only neurotransmitter for skeletal muscle, okay? All right, that's in mitochondria. Don't worry, everything that's blue is called Nissl, which is basically, rough ER, but it's called Nissl when it's blue, because that's what Dr. Nissl called it when it's stained nice and blue, okay? Now, just for testing purposes, whether it be chapter test or lab, we call the surface of the Bouton the presynaptic membrane, and we call the surface of the Bouton he talks to, or the nerve he talks to, or the muscle he talks to, as the postsynaptic membrane. Got it? After the synapse and before the synapse. Got it? So, on a chapter test, what could I ask? Where would I find receptors? On the postsynaptic. Where would I find the neurotransmitter itself? In the pre, because it comes from the pre and goes to the post. Does that make sense? Then it gets reuptaked. We cut it in half and put it back up. Right? Have we already had test 3?
Yeah, we've already had that.
Yeah, okay. All right. And then your typical neuron, which we showed you, I think we can identify a nucleus, right? And a little nucleolus on the inside. Remember, the thick part is the axon hillock. All of these white extensions coming out, which have been cut, are the dendrites. And all of these little brown things coming from another neuron are the boutons from another neuron. However, I need you to know for your test, when I have a bouton talking to a dendrite, that is a axodendritic synapse. If it's talking to another axon, axo-axonic. If it's talking to your body, axosomatic. Because the bouton is always axo something. It's always from an axon. I wonder what those dudes are doing up there. Hi. Okay. Are you good? I don't think I have enough, time to have you guys memorize, smooth ER, rough ER, Golgi, mitochondria. You know what I'm saying? But do you understand the main things? Okay? All right. You can tell the rough ER because it's got what? Little dots on it. Yeah, it looks rough. Like sandpaper. All right? That will definitely be on your test. What's the gap? Okay, what's the covering on the outside? No, the myelin is covering this. What's the protective cover? Remember? Around one muscle is endomysium. Around one nerve is endoneurium. Right? Okay. That's the actual axon. See, here's more Nissl. Alrighty? That's the nucleus of the Schwann cell who made all of the myelin sheaths. What do I call the glial cell that makes myelin around 100 axons in your brain and spinal cord? Oligodendrocytes. The ependymal cells are the ones that push the fluid in your ventricles. Okay? Which will be on the next test. Then the parts of the brain.
You said oligodendrocyte?
Yeah, oligodendrocytes. Now, see, here's a typical cell, the one I actually showed you in class, where we don't distinguish between the rough and the smooth ER. It's just all stained blue, and so we call it Nissl. Okay? But I still have my boutons, I still have the axon hillock, I still have my dendrites sticking out. Okay? Still have a nucleus and a nucleolus on the inside. All right. Now, I'm not going to get all this bent out of shape on this test, but on the next test, you'll have to know it. What do I call all the nerves that are coming in to the top side? Sensory. All the ones coming out from the bottom? Motor. What do I call the ones in the gray matter in the center? Interneurons. Because we drew it on the board and talked about it last week. Okay? The more detailed stuff, we'll do that later. Okay? But just your typical in, out